This solution looks completely ordinary at first. Tad bit yellow, but ordinary. That is until it turns into this dark blue color. This happens because before each step, there needs to be a reaction that happens before it. This is exactly what happens in life too. Inside a cell, there's chemical processes going on step by step. This is called the chain of reactions. The thing is, each step needs a helper.
In the 1940s, Beadle and Tatum used the mold neurospora crassa to show that one gene makes one enzyme. This was called the one gene, one enzyme hypothesis. Later, scientists found out that not all proteins are enzymes, so it became the one gene, one polypeptide hypothesis.
A gene is an instruction. Those instructions are used to make polypeptide, which is a chain of building blocks. That polypeptide is either folded up to make an enzyme, which is like the catalyst in a reaction, it speeds up the reaction, or it's used to make different types of proteins, such as like hormones, fibers. The iodine clock reaction, which is the experiment I showed in the beginning of the video, think of the dark blue color as the polypeptide, and think of the chemicals themselves as the genes. If a gene were missing, that means I wouldn't have a certain chemical. I don't get that dark blue color. If the genes were mutated, think of the chemicals being, if I had too much of a certain chemical or too little of a certain chemical, I still wouldn't get that same dark blue color. This happens the same thing with genes. You're not going to get what you need because it cannot happen.
The one gene, one polypeptide idea is important because it shows how DNA makes proteins, the molecules that drive life. And that's why this idea still shapes biology today, because at the end of the day, everything happens because of one gene and one polypeptide.